right now I want to talk a little bit about how an electrophoresis chamber works, how the auger works, the augurose, and why we have to add this buffer stuff to make this all work. And so hopefully you remember from physics that when we run electricity through water, we're going to have a process called electrolysis, separating the water molecules. That's going to cause an acid and a base to form, so thereby changing the pH of the solution. We know from our chemistry lessons that buffer is a chemical that resists change in pH. So when you add the buffer here and then you run the electricity through the chamber, it helps prevent the acid and base from forming too quickly, which could denature your DNA. And you don't want to ruin your samples because the pH is wrong. And so hopefully you know that we're going to load our samples into these wells, and whether they're dyes or they're actual DNA samples, they're going to be negatively charged. That negative charge in real DNA comes from the phosphate molecules, and we're going to run the electricity through here. The negative end is the black end, the positive end is the red end, and so once we start running the electricity through here, this negative current is going to repel and push away the negative molecules of the dye or of the DNA sample toward the positive end of the chamber. And if you have samples of different sizes, you're going to see a variation in the distance migrated of the bands. You want to always make sure that your wells are facing the negative end. It's one of the most common problems we see with students, that they put their gels in backwards and then they can't find their DNA because it's run off the wrong end. I'm going to show you how to do what we call a wet load. You can dry load if you've ever seen that before or used it yourself. But the easiest way, I think, for students to load is what's called a wet load. And to do that, we're actually going to pour a buffer into our chamber, and then the dyes are more dense than the buffer, and so they'll sink down into the wells. So I'm going to go ahead and fill my chamber with 1x TAE, making sure to completely fill the chamber as well as several millimeters over the gel. Depending on the brand of chamber that you're using, you may need anywhere from 250 up to about 600 milliliters of buffer, and that's just going to depend on your chambers. If you're not sure, you can always pour in some water first and see how much you needed. Okay. So this is a set of dyes that I actually prepared myself using common loading dyes such as methylene blue, xylene orange, and then a little bit of glycerol to make them heavy. And so we're going to go ahead and I'm going to load these samples and then start this run so that you'll be able to see the results of just a simple dye lab. There's no biological material in this, and so the gel and the dyes can be thrown straight in the trash when you finish. Okay, I'm going to take my pipetter, set it to 20 microliters, go ahead and get a tip. And then I'm going to load 20 microliters of each sample. Before I do, I'm going to give each sample a small flick so that it's ready to go. Okay, again, there's glycerol in these samples, so they're heavier. They're going to sink down into the wells. Okay, and so each of these dyes is going to be representing some DNA evidence found at a crime scene, and then the samples of the suspects. So I'm going to go ahead and use this purchased set of dyes. You can see here we have this little strip of tubes that are sealed together, and each one is labeled with a sample number A through G. Again, this is another crime scene lab. This set, it gave us enough for 6 groups. It also came with stain, the augurose, the buffer. It came with everything I need for about $60. And so if you don't know how, or you don't feel comfortable mixing up your own loading dyes to make samples for your students, something like this is a pretty cost-effective option. So we're going to go ahead and do sample A. Again, I'm just going to pierce the pipetter right through that foil. Go ahead and pipette up my sample and load it into the gel. Okay. As you can see, I'm just disposing of my waste into a beaker. At the end of the lab period, I'll go around and clear all those out from all my students. And then we'll go ahead and get this ready to electrophorese. Connecting the lid. You can see this lid does go on a little bit differently than the other, sliding horizontally into place. Making sure to connect those leads, get them all the way connected. Okay, and then we'll go ahead and plug this into our power supply, and then make sure your power supply is off, and go ahead and connect your cables, red to red, black to black. Okay, and again, this set uses about 125 to 130 volts. Again, it depends on your equipment. So I'm going to do 140. I'm going to set my time for 30 minutes. This one requires much longer to run since it is simulating restricted DNA. Again, there are no biological materials in this kit. However, because it's simulating DNA, it does run a little bit differently than the homemade dyes. So again, 30 minutes, 140 volts, and run. And again, in our chamber, we should see that DNA spa. At each of the ends, we should see bubbles forming as the chemical reaction of electrolysis begins to occur. And electrophoresis, again, the voltage may vary. Make sure you read the instruction manual that comes with any purchased lab. This one is about 150 volts for about 40 minutes. So this one's a little longer than if you do the homemade dyes. But again, your students will get to stain and destain the gel so they can have a more realistic experience than just making homemade dyes.